We turn now to audition or hearing, and we're going to follow the same route again. We're going to look first at the primary sensory surface, which is the basilar membrane in the cochlea in the inner ear. And we're going to look at the route to the primary sensory cortex, which lies in the temporal lobe, just above the ears here. Now, hearing is, of course, concerned with sound, and sound is generated through vibration or passed on through vibration. Something happens in the world that sets air vibrating. Those vibrations reach the pinna, the outer ear, modulate the eardrum, causing it to waggle. Those vibrations are passed through a series of bones to a little window, which causes fluid in the cochlea to waggle in sympathy, as it were. Now, any sound is made up of a whole bunch of different frequencies, from low bass frequencies to I can't do high notes, to high treble frequencies. And we measure frequency, of course, in terms of vibrations per second, or hertz. If something vibrates below 20 cycles per second, we don't perceive that as a pitched sound. We hear discontinuous events. But once those vibrations reach 20 times a second or faster, we begin to hear a pitch, a low pitch up to a high pitch. And for a young person, the range of frequencies that we hear extends from about 20 hertz to about 20,000 hertz. We looked at the psychophysics of this before. We can understand that roughly as a log scale. And here is an audiogram, which is a map of the sensitivity of that a person displays to sound at different frequencies. The green line shows the threshold of hearing. If the intensity doesn't get above the green line, you don't hear anything. And you can see that that's not a flat line. Rather, we're very sensitive to sounds between about 200 and 10,000 hertz, and markedly less sensitive to very low frequency or very high frequency sounds. The blue line at the top indicates intensities you really don't want to exceed, because after that you're going to start damaging the apparatus in the ear. If you go beyond that again, you will eventually reach the threshold of pain, as when you're locked in the bell tower with Quasimodo in Notre Dame. What you can see is that the vibrations typically found in speech, in the sounds of speech and in the sounds of music, are very well matched to the sensory capacities of the ear. The way in which speech works has learned to exploit the sensitivities that we have in audition. Now, there's the outer ear, the pinna, and you can see that there's an awful lot of structure inside, buried deep inside. We can see the eardrum there, and then there's a complicated set of bones, the hammer, anvil, and stirrup, which pass on the vibrations into the cochlea. Also attached to the inner ear are the three structures of the vestibular system. These are semicircular canals. They have nothing to do with hearing. They are concerned with orientation, acceleration, and they contribute to our visual understanding of the world. We are concerned here with the cochlea. And we're going to blow up that picture. The cochlea is shown roughly life-size here. It's a very small organ. If we blow it up schematically, it looks like this. You can see where the vibrations coming through the eardrum and the hammer, anvil, and stirrup, given their Latin names here, they push on a little window, moving a fluid called a perilymph throughout the structure of the cochlea. The cochlea has this long extended structure. In the middle of it, we will find the basilar membrane, the hair cells, and another membrane called the vestibular membrane. Outside of those structures, we have the perilymph. So the perilymph is vibrating in sympathy with the sound coming in, and that causes a differential response along the basilar membrane. Looking now in more detail, shown in the bottom left here is a tiny portion of the basilar membrane where you can see hair cells, the outer hair cells and the inner hair cells, which are attached to the tectorial membrane. Now, when the perilymph wobbles due to the sound, that causes sympathetic wobbling at different points along the basilar membrane. And where the wobbling is most intense, those hair cells will be stretched, and that will cause the nerves there to fire. So the outer hair cells here are playing the role of the photosensitive cells in the eye, and this is the point at which transduction into nervous energy occurs. And if we simplify, because the cochlea is very complex, if we simplify the cochlea like this, we can see it is a rolled up spiral, which if we stretched it out, would be basically a straight line. And what's shown in the diagram on the right is the differential response to different frequencies. So high frequencies come in, and it is the first part of the basilar membrane that is excited by these. Mid frequencies excite another place, and low frequencies excite another place. So what we've done is we've turned the frequency content of sound into a spatial layout, much like the retina. This is a one-dimensional spatial layout organized by frequency, not a two-dimensional spatial layout like the retina. But it's a one-dimensional spatial layout, and that's what we're going to map now to the primary sensory cortex. And the plumbing, as usual, is appalling. The ear is right beside the temporal lobe, but that's not how the plumbing works. As you can see here, I've only indicated some of the complexities. There are several way stations between the cochlea and the auditory cortex. There's crossings and mixings of signals, side branches going off to other organs, and so on. It's complicated. It's not a simple feed-in system. Nonetheless, when we turn our attention to the primary auditory cortex in the temporal lobe, what we find is a spatial layout organized by frequency. This is a tonotopic map. We saw previously that the structure of the retina is preserved with some shrinking and stretching as a retinotopic map in primary visual cortex. Now we're seeing the same principle operative in audition. That is, we have a spatial map organized by frequency in this case, which projects to a primary sensory cortex, which preserves this mapping. So that's an important commonality between vision and audition, and as we'll see in the next video, touch shares this form of organization also, whereas smell and taste and proprioception and kinesthesia do not. Now, there are very many important differences as well, of course, between hearing and seeing. One point that immediately springs to mind is that the eyes are directed towards one part of the world or another on a whim. We decide to look up, down, left-wise, sideways, but the ears just seem to be always open. We can do this kind of directional hearing, but we don't do it as a role. And we can close our eyes, and you can't close your ears. You can stick your fingers in your ears, but you can't close your ears. So the ears seem to be always on, in a sense, which encourages us to think of this as a pure input modality. Now, we have a lot to learn still about hearing. I don't just mean in this course, I mean the science of hearing is very underdeveloped. One thing we have found out is that the sensitivity of those hair cells in the cochlea is actively modulated at all times. And indeed, there are as many fibers going from the brain to the ear as there are going from the ear to the brain. This means that the ear, the basilar membrane, this is an active organ which is constantly participating in the business of sense making. It is not just an input channel, and the ear is an awful lot more than a hole through which the sound gets in. Now we'll turn our attention to touch in the next video.